Welcome to episode 2 of the Antimicrobial Agents and Chemotherapy podcast. The AAC podcast is the official podcast of the journal and features discussions with investigators and opinion leaders on the latest cutting-edge research and events related to the treatment of infectious diseases, including aspects of antimicrobial resistance, epidemiology of multi-drug resistant organisms, pharmacology of antimicrobial agents, and susceptibility testing, among others. I am Cesar Arias. I am professor of medicine and microbiology and molecular genetics at the University of Texas Health Science Center at Houston. And I have the honor to be the incoming editor-in-chief of Antimicrobial Agents and Chemotherapy. Welcome to episode 2, and we have a very important and I'm very pleased to have the guest we have today is Dr. Lou Rice. Dr. Lou Rice is the current editor-in-chief of Antimicrobial Agents and Chemotherapy. He is the Yukolsky Family Professor and Chair, Department of Medicine, the Warren Alpert Medical School of Brown University, and Physician-in-Chief, Rhode Island and the Miriam Hospital. Lou, welcome. It's a real pleasure to have you here.
Thank you, Cesar. It's a pleasure to be with you.
Thank you very much. Our podcast today is entitled AAC Past, Present, and Future. And it's a real honor now to share this with you because you have a perspective that's very unique in the field. So I want to take this opportunity to thank you for all the services for the journal in the past 10 years. And also start by asking you a very simple question is, how did you end up doing research in antimicrobial resistance?
That is a very simple question. Unfortunately, it doesn't have a particularly simple answer, so I will take it from the beginning. It wasn't an obvious thing that was going to happen to me. I was a philosophy major in college. I didn't want to do science. I had no interest in science. Actually, mostly what I wanted to do in college was play football. When before my senior year, I had to think about what I was going to do with my life, and I had spent my summers as an orderly in a nursing home. So I thought, well, I'm not going to be a philosopher. I'm not smart enough for that. The people in the philosophy department at Harvard didn't seem like people I necessarily could easily hang out with anyway. So, and I didn't want to be a lawyer. So I thought, well, I could try medical school. Fortunately, and then I took classes and managed to go to medical school down at Columbia, which was a really terrific experience. In fact, I have to say, there was a wonderful teacher by the name of Glenda Garvey who taught me in my second year all about infectious disease. And honestly, the impression I had when I learned about all the bugs and the antibiotics was, what kind of nerd could possibly want to do this for a career? There's just, I could think of nothing worse. In fact, I thought I wanted to be a gastroenterologist. However, when I got on the wards, turned out all a gastroenterologist wanted to do was put scopes in people, which I didn't have any interest in doing. And the infectious disease people were the ones who were asked to talk about all the interesting cases. And it seemed like detective stuff, and it seemed fun. And so I thought, well, it's going to be a bear to learn all this stuff, but it might be fun to use. But I never thought I'd do research. And then I ended up at Bellevue Hospital for my residency right at the beginning, actually the end of the beginning of the AIDS epidemic. And it was truly remarkable because, sort of like COVID, only it was more slow growing, but it was a time when there were a whole lot more questions than there were answers. So by the time I finished my residency, I knew I wanted to do infectious disease. That was clear. But I thought I wanted to research AIDS. I was going to research pneumocystis. My wife was doing a residency in Boston, so I applied to places in Boston, was fortunate enough to get into the Deaconess Hospital. But the Deaconess Hospital didn't really have anybody researching AIDS. Scott Hammer was researching herpes viruses at the time. What they had was Bob Mollering and A.W. Karchmer and George Iliopoulos. And over the course of my clinical year, I kind of became fascinated with bacterial infections. And so I thought I want to do research with Bob Mollering. And so I had a few projects. They put me on a few projects. And the major one I decided I wanted to do, I think you'll find this somewhat humorous, Cesar, is I was going to determine what the mechanism of action of this new antibiotic called daptomycin was. It was a brand new antibiotic.
The circle of life, huh, Lou?
Lilly was developing it at the time, and not only was I working on it in the laboratory, and I even published a study looking at synergy between daptomycin and fosfomycin against gentamicin-resistant Faecalis strains. I was also the unblinded investigator on a clinical study, bacteremia. And so I was deep into daptomycin. I realized in the second year of my fellowship that I loved research. And then Lilly decided that daptomycin didn't work. It didn't. They changed the dose a little bit and the toxicity went up and they put it on the shelf. And so after a year, when I decided I really wanted to do research for my career, I had nothing. I was bereft. Wow. And Bob Mollering took pity on me and said, Lou, we've got these strains from Boston Children's Hospital, and they're Enterococcus faecalis, and they make betalactamase. Why don't you go over and work with George Jacoby in his lab at Mass General and learn how to do a southern blot and tell us what plasmid it's on? Well, I had also in that year done work in Bob Mollering's animal lab, where I had worked with a woman named Sandy Willey, who left during the year to become the infection control officer at Yeovil Hospital, which was a chronic care facility in Cambridge. And while I was working in George Jacoby's lab, Sandy called me and said, you know what? We got a bunch of bugs here. They're Klebsiellas, and they're resistant to ceftazidime. This was 1987. And said, are you interested in those bugs? Well, George Jacoby, of course, was Dr. Betalactamase. He was an expert. And as it turned out, I found out with the Enterococcus that the betalactamase wasn't on a plasmid, it was in the chromosome. So from the ashes, and we reported, I think that was probably the second outbreak of ESBLs we reported, that was reported in this country. There was both TEM12 and TEM26. And on top of that, the examination of the enterococcal chromosome kept me funded for 10 years, trying to figure out that 65 kilobase transposon piece by piece. So all of a sudden I had a great career. And the next person who was important in it was the person who recruited me out to Cleveland, David Schlaze. He recruited me because he wanted someone to partner with. And he taught me some very important things, like, buddy, you better get funded or these people are going to make you do a lot of clinical time. So he taught me how to write grants. And then it was off and running. As you know, it was an easy field to have fun with because, you know, there's just a lot of colorful personalities other than the ones I've talked about. I know you've worked with Patrice Corvalin. I mean, he's a very colorful personality. I've really come to become very fond of Patrice, but I learned early in my career that if you wanted a career, the smartest thing to do would be to figure out what Patrice was interested in and do something else, anything but that. He was so effective at what he did. But people like Stuart Levy, John Quinn, Virginia Villegas, all these incredible characters, and then working with some amazing trainees. Robert Bonomo actually was a fellow in the lab of Dave Schlaze when I got there, and David Patterson came and worked in his lab. You know, each one of these people have contributed so much, not only to my intellectual growth, but just their warmth and their their decency has made it a great field to be in. So that's a long answer. That's how I get into antibiotic resistance. And I've never regretted it for a second.
Thank you, Lou. That's a wonderful answer because you're mentioning so many superstars in the field that is just staggering. It's just amazing. And I wanted, with that knowledge,
Cesar, I would be remiss, honestly, if I didn't mention the person I've always considered my sibling, my sister from the laboratory of Bob Mullering, and that's Barbara Murray. She has been extraordinarily helpful and a wonderful friend throughout my career, and another one of those very colorful, interesting personalities.
Yeah, I think I've heard of her. So you've been able to, as editor-in-chief in the last 10 years, and all your experience previously, I guess the 20th and the 21st century now, to see the evolution of the field on many years. So how would you describe what have been important sort of events in the field that have pushed the field forward, or has challenged us to just come up with something important? And of course, now as multi-drug resistant has become such an important topic in the field and has been elevated as a major public health problem, where can you give us your perspective on how this evolved, because you've been part of it in a very major way?
Well, I think if there's one thing that I think has revolutionized and changed entirely the field of the study of antibiotic resistance, and frankly, probably all of science, occurred when I was a fellow, and that was the first descriptions of the polymerase chain reaction. People, it's such a common thing everywhere in science now. It's interesting to think back to the fact that I remember talking in the lab with George Jacobi about this new technique, and did he think that it was going to make that much difference? And he said, well, of course. Anyone who's ever tried to clone a gene, especially one that doesn't necessarily confer a phenotype, knows that was hard. I mean, the good thing about it was if it took you 6 months to clone the gene, people understood. And, if you only published 2 papers a year, people thought that was good. But now everything is hyperspeed because there's no more secrets in the genome. That's probably the other major advance that I think has changed the world. Is what used to be very difficult to get. It used to be difficult to get the genome sequence. For a long time, there were no enterococcal sequences published. Down there in Texas, you were among the first that were really interested in that sequencing. I actually got a grant from NIH to sequence one of my Ephesium strains, and they were going to sequence it for me. And it took me 9 months. It took them 9 months to sequence it. So here we are now where, people who are smart and they're clever, they can get so much information. I mean, readily, freely available on the internet. There's probably 1,700 enterococcal sequences now. I don't even know. And as long as you have somebody who understands how to use the programs and do bioinformatics, you can get a lot of information. Honestly, I was serious when I said it took me 10 years. And I got 10 years of funding to figure out a 65 kilobase composite transposon in the enterococcal genome. And it was an interesting finding because it was a composite of staphylococcal and enterococcal sequences. But right now, that's paying $100 for a sequence and, getting some bioinformatics time. And 2 weeks later, you got it. There it is. Yeah, maybe the insertion sequences will give you some trouble, but you'll be able to figure it out. And so I guess I'm kind of, I miss those days because you had to use your imagination. And a lot of times you you were benefited if you weren't well educated. So I was not well educated in the sciences. I was a latecomer, so I knew a lot about a small area, but I didn't know enough to know that I shouldn't think about the things that I was thinking about, and it turned out some of them were true. So it certainly benefited me. But the questions that people can ask now are really big questions, and they require really big brains. And so, it's not my field to think about all these genomes and how everything works together and the systems aspects of it. That's certainly where things are going, and I applaud it. I think that's where it needs to go. I think we need to understand how bacteria interact with each other, the signals they send to each other, how they live together. We need to hopefully be able to use that to not only try to inhibit the ones we want to inhibit, but to try to support the ones that we want to support. But that's for people who are really smart and have a lot of training and knowledge. So I like reading their papers. I admire them. But I was more at home in an era where you had to use your imagination to explain a small finding, and where people would excuse you if it took you 2 years to figure it out.
That's a wonderful story and view. Many people who knew that I was going to do this podcast with you wanted me to ask you about escape. I mean, it's something that has your name all over it, and I guess the only thing that I would regret is it wasn't published in AAC. I would have loved to have been published in AAC, but everybody said, ask Lou, how did he come up with escape? And did he think that acronym would have put that weight that now is everyone talking about resistance, and basically changing the feeling, the attitude of funding that made a huge difference. And I include myself on that, and I want to thank you for that. But people want to think and wonder, how did you come up with that?
I'll be happy to tell you that story, but before I do, I want you to I want to make an important point, and that's that I was a latecomer to the game. If you can go back and look at, I think it was Nature or Science, there's an article published that talked about the first meeting that was pulled together to talk about antibiotic resistance with NIH, and it was in the 1990s. And it was pulled together by David Schlaze, Stuart Levy, and Gordon Archer from Virginia. And they brought the NIH people together and they tried to convince them the importance, and it didn't work. And they did another one 3 years later, and it didn't work. And there were others. George Jacoby was also involved. There were just huge people in the field who were dead set on making sure that the NIH had to pay attention to this. So I grew up watching them. And it was a matter of circumstance that I happened to be working for the infectious disease societies. I was chairing their research on resistance working group. And one of our charges was to find out from the NIH, what is your research portfolio on antibiotic resistance? And how can we help you expand it and prioritize it? That's what we wanted to do. We wanted to help NIH with that because we felt like they may not have the expertise inside to do it. They didn't want to open up their portfolio to us, but they said that the person who was responsible for resistance at the time, one of the people was Kent Peters, that he and Tony Fauci would write an article talking about the NIH portfolio on resistance. And so they wrote that. And because I was the chair of this research on resistance working group, Marty Hirsch said, I would like you to write a commentary on it. I said, sure, I'll write a commentary. Just send me the article. Well, I read the article, and the article was, I'm sure it was all true. But it's like one of those questions that we used to get on standardized tests, where one of the options was true, but unrelated. The article talked about the millions and millions and millions, hundreds of millions of dollars they're spending on antibiotic research. And that included all sorts of things like tuberculosis, malaria, HIV, and it was all lumped in. And all of it was true. There was nothing about it that was untrue. But you couldn't sift out what they were spending on antibiotic-resistant bacteria, which was the problem that we wanted to find out about. And I got irritated, and I think I sent an email saying probably something ill-advised. And Marty Hirsch sent me an email saying, this is an accepted article, and you're going to have to, either you're going to write a professional commentary or it's not going to be published. And I said, okay. At first I thought, well, I'm just not going to do it. And then I thought, well, when are you going to have an opportunity to do this? And I was trying to figure out how to make the point that what we need is research on Staph aureus and Enterococcus and Klebsiella and Pseudomonas. And I said, my God, I get bored and fall asleep by the time you get to that fourth organism. How can I keep referring to this in a way that's not going to get so boring that people are going to stop reading? And literally, I was laying in bed one night, and the letters were just floating around in my head. And aside from the misspelling with a C with a K, I thought, wow, that's if you put, Enterococcus, Staphylococcus, Klebsiella, Acinetobacter, Pseudomonas, and Enterobacter together, which is really probably 80 to 90% of the resistant bugs we really worry about, you can actually make a reasonable proximity of escape. So I made the headline, NIH portfolio on resistance, no escape. And I tried, in a polite way to say, what we really need to know and what we really need is money put into these bacteria, which are important in the hospital. I had no illusion that it was going to do anything but be in that title, but it seems to have been taken up and people use it as an acronym now. And I guess I'm proud that I was able in, 2 o'clock in the morning to come up with that.
The NIH responded to this as well. I think, what was the response to NIH? Because then you chair, the DDR group was formed and you chair that group. And it's very interesting to see what was the response of the NIH after that.
Well, the DDR was actually formed a few years before that. That was one of the early things that NIH did. I think that I had been a member of BM1 at the same time they formed DDR. So I split off to DDR from BM1. And any of us that had been in BM1 had become frustrated that, resistance just wasn't going to be funded out of that. Not enough people understood it. And it sort of had become a bit balkanized into the E. coli group, the chlamydia group, the TB group, the spirochete group. And so I was very grateful when they formed DDR. I think it was great. And I think it's still a terrific study section for us. And so I'm very grateful for that. I think the real difference, or I think one of the most important things they did, and I do credit Tony Fauci for this. I think he listened, and he created it. Was the I'm having a senior moment. Why am I having a senior moment? Well, the clinical studies group that Vance Fowler and Chip Chambers are running.
ARLG.
ARLG. I think that was a real positive thing. If there's one thing I could say that came out of all that was really important. One was a fair amount more funding. They started to reinterpret what bioterrorism requirements were and gave a little bit more of that to antibiotic resistance. But I think it was the important thing was the ARLG, and so I hope they give that more funding. because I think that has done a lot. It's asking a lot of important questions. And more importantly, it's encouraging young people to research in this field, which is what we need.
Yeah. So in all these processes that you've been, from the start, so can you tell us about the role that AAC has played in disseminating antimicrobial research?
Oh, I think I certainly, well, I may be a little bit biased, of course, but I think AAC is the premier journal for antimicrobial resistance in the world. It was before I became editor-in-chief. I think it still is, so I'm glad that I haven't been associated with it lessening its profile. I think it's been really superbly responsive to changes in science. Back in the 1980s, many of the studies were susceptibility studies. And that makes sense because at that time, there were lots of new antibiotics coming out. And there wasn't a whole lot of scientific technology to look at mechanisms, et cetera. When Bob Mollering was the editor-in-chief, he really, I think he really upped the scientific profile of it. He really wanted more basic science, and George Jacobi followed with that. And George Iliopoulos the same way. It was really, they were really smart people, and they pushed it in a lot of different directions, particularly in a molecular direction. I think in the 10 years that I've been there, I don't think that I have pushed it in a more molecular direction. I think I've certainly watched the science move in that direction, but I noticed that recently in researching an article, sorry, Melissa, I will try to get that article that I want to write on the last 10 years, or last 20 years of AAC, myself and George Iliopoulos. I looked at what the different categories of articles that were published from 2000 and 2001, and then 2018 to 2019. And there's some interesting trends. We have an epidemiology and surveillance section, and now we didn't even have a section like that then. So there were no epidemiology papers published in AAC back then, but it was somewhere about 8% of the articles in the subsequent the 2018 and 2019. There are less susceptibility articles now. There are slightly more pharmacokinetic, pharmacodynamic articles. Clinical therapeutic section is more robust than it was, and I think this just reflects what's happened in the antibiotic world, in that there aren't so many brand new antibiotics coming out, and beyond that, we have well, of course, your brilliant suggestion. When you wanted AAC to consider making a challenging clinical cases article, and then I pulled the classic medical school professor. I said, okay, you want it? Why don't you come be the editor of it? And that's just been hugely successful. I mean, it's been a real game changer for us, as will, I think, your new ideas to have AAC be the repository of review articles on all new antibiotics. I think that moving the journal more in that direction is going to be a huge advance. So I think AAC responds to the time, it responds to the science. I periodically have to review things, and I go through other journals for over a year, and then I'll review all AAC as well. I don't think any other journal that addresses antimicrobial chemotherapy comes even close to AAC in terms of the quality of science. Final thing I'll say is it's all thanks to our editors. We're fortunate that AAC is such a good journal, has such a good reputation, that true huge giants in the field are willing to come on and be an editor. The editorial group that you are taking over as editor-in-chief, it is a great group. and you've added some terrific people. So it's a living, breathing thing. It's going to change. I can't predict exactly how it's going to change. Science will determine that. And you'll find that as editor-in-chief, you can it's a big ship, and you can't exactly turn it on a dime. But over a few years, you'll be able to put your stamp on it. I really look forward to that, because I think 5 years from now, it's going to be even better.
Thank you, Lou, for your kind words. Just a final thought from you. What would you say to a young person entering the field? What will be an important thing that you will tell a young scientist, physician scientist, or a PharmD entering the field of antimicrobial resistance? What would be your main recommendation?
My main recommendation is when you're young, have an open mind. You never know what question is going to turn out to be really interesting. You may think something is going to be really interesting, and another thing might not be. Once you get almost any area of science, once you get into it, you will find that there are many interesting questions, and asking questions is really, really important. If I had an opportunity to get into an area that I thought was really interesting, and that somebody else said, oh, this is what the future is going to be, or go into an area where there was a terrific mentor, a generous, decent mentor who was really smart, but not an exactly exciting area, I would go into the second area. Because now we all think our own experience, is good, but I can't tell you how many people, when I went to work in Bob Mollering's lab, said, why are you researching antibiotic resistance? Only the pharmaceutical companies care about that. And if you're going to research antibiotic resistance, why are you going to research the enterococcus? Because only Bob Mollering cares about that. Well, I said, I don't care. I think he's a wonderful mentor, and I think there's a lot of interesting stuff here. And as it turned out, enterococcus became a really important subject, and is even able to attract people like yourself. It was a great field to go into, but when everybody in the world thought AIDS was the only thing to go into, enterococcus was not popular. So find a good mentor, listen to that mentor, work with that mentor, and then once you become successful, recognize that science only advances through generosity. And be generous with people, be generous with your knowledge, be generous with your strains, be generous with your techniques. You will find it's a wonderful world. The people 30 years later in antibiotic resistance are no less interesting and no less wonderful than they were when I got into it. So I think it's a great field. I think we need it. And I don't think anybody will regret it. Plus, if you're a physician, and I'm not saying only physicians should do it. That's certainly not true. But if you're a physician, you have the wonderful ability as an antibiotic resistance researcher to be on the wards and to absolutely feel like you're working with your relatives. When I look at a susceptibility profile, I know how they're doing it. And I can explain to people how they're doing it. And so that is such an amazingly practical way of applying science that you often don't get in other areas. So especially for the physician part, it just is great fun to be on the wards and training young people, because their eyes just open up wide if you can explain these things in a way that they can understand.
Well, that's been a wonderful conversation, Lou. As I'm going to put my head as incoming editor-in-chief, and I want to thank you from the bottom of my heart, and I'm sure on behalf of the American Society of Microbiology, for these wonderful 10 years to keep the science going, for your wisdom and support to all of us. I count myself on that. and I hope to keep counting on you in the future. And thank you very much for your time.
It's been a great ride, Cesar. It's been wonderful working with you. It's been wonderful working with everybody at ASM. I have enjoyed thoroughly it's actually been 20 years, only 10 as the editor-in-chief. I've enjoyed the entire time. I will miss it, but boy, I'm looking forward to some free time.
Thank you very much. We conclude now. This is Cesar Arias, incoming editor-in-chief of AAC in the AAC podcast. Thank you for your attention.